Hello, everyone. Firstly, I would like to thank Professor Woding and the organizing committee for including me in this exciting symposium. My name is Hatice Altug from EPFL Switzerland, and in my talk, I will show how in my research group we are exploiting nanophotonics for biosensing. In simple terms, biosensor is an analytical device that can detect and analyze biochemical substances. The schematic here on top shows its main components. It has a transducer, which can get functionalized with biological receptors. And this component can be adapted to detect a wide range of different target analytes, such as drugs, proteins, DNA, viruses, etc. The transducer response can be diverse, ranging from simple yes or no indicator, as in the case of rapid tests, to all the way quantitative detection, analysis of interactions, or even extraction of molecular confirmational information. And because of this diversity, biosensors have a wide range of applications, including life sciences, pharma, diagnostics, environmental monitoring, food safety, and security. And currently, biosensing research is particularly focused on diagnostics. And this is because diagnostics is the first step in medical care, and any follow-up treatments are based on diagnostics. But today, diagnostics face serious challenges. It's time-consuming. It typically requires large sample volumes, centralized labs, expensive instrumentation, and trained personnel to run such instrumentation. And because of that, it's also not available to everyone, which is an issue at resource-poor settings. So that's why looking ahead, we need to consider completely different approaches for medical diagnostics. In particular, with the emerging healthcare needs and initiatives such as global healthcare, point-of-care diagnostics, and personalized medicine, we need to develop revolutionary bioanalytical tools that can analyze almost any sample on the spot whenever and wherever we need. To realize such powerful sensors, clearly we need scientific and technological innovations in order to have some desired device specifications, which can be, for instance, sensitivity, low cost, portability, intelligence, on autonomous operation, among others, depending on the application focus. In fact, an appealing vision for the future is that such next generation biosensors will be everywhere. They will, for instance, serve at the nodes of Internet of Body and Internet of Things towards smarter systems. In fact, here I would like to invite you to check our recent review papers, which highlight some prospects for nanophotonic sensors. In this context, the mission of my lab is to develop science and technology for powerful optical biosensors that can have impact on areas ranging from basic research in life sciences to real-world applications in diagnostics. And in our research program, we leverage several key technologies, including nanophotonics, nanofabrication, microfluidics, surface chemistry, imaging, and data science as well. The research in my group, I categorize roughly into three main directions. The first one is on the development of point-of-care diagnostic tools. Here we introduce, in particular, low-cost, portable, rapid, and multiplex microarray-based technologies. The second direction is on novel mid-IR spectroscopy techniques, and here we leverage mainly chemical-specific detection and confirmation-sensitive detection. And the third one is on the development of new technologies that can enable one-of-a-kind measurements on living cells, in particular, single cells. So firstly, I would like to focus and give an example for a point-of-care biosensor. So here on the photo, I show one of our recently developed portable and low-cost nanophotonic biosensors that can enable rapid and digital detection of biomarkers for diagnostics. The device is low weight and uses low-cost and over-the-shelf LED and cameras. We have a nanoplasmonic microarray chip, which can enable multiplex detection. The biochip is based on plasmonic nanohull arrays, so that's shown here schematically. And when we illuminate light on this nanostructure, it can resonantly transmit light at a specific wavelength due to the excitation of propagating and localized surface plasmons. And this transmission is called extraordinary optical transmission, or EOT in short. In our 2018 paper, we showed that this a signal can be locally suppressed with the binding of properly sized gold nanoparticles near the nanohull arrays, or inside the nanoholes. And with this plasmonic mechanism, we demonstrated that we can digitally visualize individual sub-wavelength nanoparticles at a high contrast using a standard CMOS camera. To utilize this effect for sensing, what we do is we coat the nanoparticles with detection antibodies and use them as a probe in a sandwich assay configuration to digitally image on the sensor surface the binding of a target analyte, for instance, this red one. Our bioassay principle is simple and single step. What we do is we first mix the conjugated nanoparticle solution with the sample, which could be serum sample, for instance. Next, we load this mixture onto the biofunctionalized nanohull array chip, which can be then also embedded in a cartridge. Then we insert this chip in our portable reader and start imaging the binding of nanoparticle labeled biomarkers in real time, as shown here. And we can count them quantitatively. We show that our nanotechnology has an excellent sensitivity with limit of detections that can go to few tens of picograms per milliliter and have a high dynamic range of on the order of 4 orders of magnitude. We fabricate these plasmonic chips at low cost using wafer scale deep UV lithography. The photo here shows a patterned 4-inch glass wafer, which typically yields around 50 1 by 1 centimeter square nanoplasmonic chips that we use in our platform. For application, we choose sepsis, which is body's uncontrolled inflammatory response to infection, and it affects more than 30 million globally, and it is the most expensive hospital treated condition. The survival chance from sepsis drops about 8% in each hour until the treatment begins. So that's why rapid diagnosis of sepsis is very important. We tested our portable sensor at the Valdebrand Hospital in Spain using patient samples, as shown here, some examples from a sepsis biobank. In particular, we tested 2 sepsis-related inflammatory biomarkers, that's CRP, C-reactive protein, and PCT procalcitonin, directly from patient serum samples. And we got excellent performance comparable to gold standard clinical assays such as CLIA. And the advantage here is that we strikingly maintain a very short time to result with less than 15 minutes, which is quite important and an advantage compared to gold standard methods. Next, I would like to highlight some selected examples from our second direction, mainly on the development of mid-IR sensors. Maybe in one slide, I would like to tell that infrared spectroscopy is a powerful label-free method, as it can provide chemical-specific identification by accessing characteristic vibrational motions of the molecules. In fact, infrared absorption signals are unique to each molecule, and they can even serve as what's called molecular fingerprints. Although IR spectroscopy is powerful, it faces some major limitations. One of them is the low sensitivity, which prohibits measurements from monolayers and to detect trace amount of samples. Another challenge is the absorption spectroscopy of water strongly overlaps with the vibrational signatures of molecules, and this imposes limitations for measurements in aqueous solutions that's water-based. Thirdly, available IR methods mostly rely on bulky and expensive instrumentation like FTIR, which limits their use for on-site applications. And moreover, it's difficult to analyze complex samples containing a large range of analytes because their molecular fingerprints can actually spectrally overlap, and it can be tricky to differentiate them. So to address these challenges, we leverage nanophotonics using a detection principle called surface-enhanced infrared absorption spectroscopy, or SERA in short. Here, IR absorption signal scales with electric field intensity, and we significantly amplify them using nanoantennas that when we operate them at their resonance wavelength. In particular, we engineer nanoantennas so that their resonance wavelength matches spectrally with the specific absorption bands of the molecules. And this interaction then leads to strong infrared absorption signals from the molecules, which then, for instance, appears as these sharp dips at the resonance location. In our recent studies, we have been extending the capabilities of SERA by engineering broadband and or so-called multi-resonant metasurfaces. And in particular, we couple them with artificial intelligence to develop powerful biosensors that can allow simultaneous monitoring of different analytes in heterogeneous samples by using chemical-specific IR signatures. And in the next slide, I will briefly highlight two selected examples. The first example is based on nanoplasmonic metasurfaces. Here we have developed significant expertise in our group to integrate media-nanoplasmonic chips, in particular plasmonic chips with microfluidics, to perform real-time SERA measurements directly in aqueous water-based solution. In our recent work, we extended our capabilities to detect different analytes from all 4 molecular classes, including polypeptides, polysaccharides, nucleic acids, and lipids. In fact, here I show their IR absorption spectrum. And as I highlighted with this colored bands of purple, green, and yellow, there are certain spectral regions which are quite rich in molecular information. You see that they are quite busy here as opposed to some regions which are flat. And in order to utilize these spectral regions containing information, we designed a multi-resonant metasurface containing 3 resonances, and we tune the resonances of this multi-resonance structure to spectrally overlap with these target bands. And here the top plot on the here shows the experimental spectrum of such an engineered metasurface directly measured in water. We tested our system using a multi-analyte bioassay that is based on liposome nanoparticles loaded with both sucrose and nucleotides. And upon binding of liposomes on the surface, what we do is we introduce melatonin, which perforates the lipid membrane. And this interaction leads to the leakage of the liposomes and release of this dual cargo, as well as formation of supported bilayer patches on the sensor surface. Since the spectral signatures of these analytes partially overlap, we use deep neural network for an effective and reliable discrimination between all of these simultaneously present biomolecules. And with our AI-aided algorithm, this optofluidic biosensor enabled us to resolve in real time these interaction events that I show you that's happening with all among these 4 major classes of biomolecules. And we do that without using any external labels. In the second example, we introduced a broadband imaging-based IR sensor based on all dielectric metasurface, what we call pixelated dielectric metasurface, supporting high Q resonances. So this top left shows this pixelated metasurface, and each pixel is composed of an array of symmetry-broken silicon nanoresonators, as shown here. And by scaling linearly the dimension of these antennas at each metapixel, we spectrally tune its resonances, shown here, throughout the mid-infrared spectrum. So here in the schematic, I show tuning over 400 wave numbers. And because there is a correlation between each metapixel and frequency, this structure in fact provides what is called one-to-one mapping between spectral and spatial information. So when we coat the sub-analyte on the top surface of this metasurface, Based on its absorption spectrum, the reflection response of each pixels get modified as such. And when we trace the envelope of the pixels, we extract the absorption spectrum of the analyte, similar to what we do with a high-resolution spectrometer. Additionally, after the analyte coating, not only the resonance peak attenuates, but also the total reflection signal from the metal pixel decreases, let's say from this gray to yellow part. And by utilizing an imaging-based readout, what we do with our metasurface is that we convert the absorption spectrum into a two-dimensional molecular barcode-like image. To demonstrate the versatility of our method for different applications, we extracted such molecular barcodes, these images, for proteins, for diagnostics applications, polymers for material science applications, and a pesticide for environmental monitoring. And as you can see, all of these barcode-like images are unique and different from each other because the underlying spectrum, the absorption spectrum of each sample is distinct. Strikingly, our imaging-based detection method can be implemented using a broadband light source and a detector without the need for spectroscopy or maybe like moving mechanical parts and tunable lasers. So in this regard, it really paves the transformation from lab-based mid-IR spectrometers to miniaturized devices like point-of-care biochemical sensors, which is something we are looking into. And so from this perspective, we also believe this novel device concept can also open up new horizons for AI-based molecular barcode analysis. To adapt such nanophotonic-based nanotechnologies in real-world applications, there is also need for low-cost and high-throughput nanofabrication methods. For this purpose, we also introduced recently a CMOS-based compatible manufacturing method for wafer-scale nanofabrication of functional mid-IR metasurfaces. The method is based on thin transparent membranes such as aluminum oxide, which can provide over 90% transmission efficiency over an impressively broad spectral window, as shown here, that can span from 2 micron all the way to 20 micron. And remarkably, this metasurface, these membranes can have also an ultra-low effective refractive index down to 1.04, which results in almost optically free-floating metasurface films in air. We use the method to realize larger and high-performing meta lenses, high transmission by refringent metasurfaces for polarization control, as well as from sensing perspective, interestingly, a CMOS-compatible plasmonic metasurfaces based on aluminum, and showed their Sarah-based sensing application. With this, I conclude the second part, and in the remaining few minutes, I will highlight one of our single-cell technologies. So in particular, within the context of immunoengineering, we have been developing several systems, and one of them is to investigate cell-cell interactions at single-cell resolution. And in particular here, we have been looking into cytotoxic T cells for anti-tumor immunity. For this purpose, we designed a high-throughput biochip from PDMS, as shown on the left, for real-time analysis of cell-cell interactions. Each chip contains more than 20,000 microvels, and each well has only 65 picoliter volume. And this tight confinement, shown here with dimensions of 35 by 35 micron, allows close interaction between a single T cell and a single tumor cell. And we load this biochip first with cancer cells, and then later with T cell, both at low concentrations with manual cell seeding, to have approximately a single T cell and a single tumor cell together in each well. And we use fluorescent microscopy to obtain time-lapse imaging with 4 simultaneous channels through 1 bright field and 3 fluorescent labels. Here, violet channel is used to trace tumor-specific T cells. Red channel is used to trace the tumor cells. And if the tumor cell is not killed by the immune cells, they stay, this tumor cells stay red, the information stays in the red channel. But if the immune cell is able to kill the tumor, then the green color is activated. And what we do basically in our technology is that we observe in a simple analogy the fighting between T cells and cancer cells in a large number of boxing rings. All simultaneously, we kind of follow their fighting. We use this biochip to observe cytotoxicity effect of immune T cells on melanoma cancer cells. So this is actually showing a live video. And this technology generates a large set of spatially and temporally resolved data because we capture the cell images from multiple channels over 24 hours within 15-minute time intervals and over a large, let's say, chip area. And this process, generates clearly a lot of data. And to process this data from thousands of bells and automatically recognize cells, we aided image analysis with deep neural network. And this enabled us rapid and quantitative analysis of single cell killing events at high throughput, where we extracted statistically relevant information on the effect of CD4 and CD8 immune cells on melanoma cancer cells. So with this, I'm at my summary slide. To conclude, emerging new health care initiatives are driving a paradigm shift in the way medical care will be provided in the future. So for that, we need to develop next generation diagnostic tools, as well as bioanalytical tools. And I believe nanophotonics will play a major role in this regard. And today I showed you several research directions that that we are doing in my lab for this need. So before ending my talk, I would like to thank my group members as well as collaborators and our funding sources, as well as I would like to thank you for listening, and I will be happy to take your questions.